Disorders of lipoprotein and cholesterol metabolism.
Definition and different types of lipoproteins. Plasma lipoprotein formation and transport in blood. Description of structure of cholesterol, its sources, precursor, function, and derivatives. And outline the types of dyslipidemias, their common causes and complications. First, we start with a definition of lipoproteins, and as their name implies, lipoproteins are clusters of lipids and proteins with different ratios between lipid and protein components. This composition is important for the structure so that they can perform their functions. The most important function of these molecules is to transport lipids in lymph and blood. As from their structure, lipids are hydrophobic and need to be bound to hydrophilic molecules to facilitate their transport throughout the body fluids, especially the lymph and blood. Other functions of lipoproteins include their structural role as they enter the structure of membranes of cells and cellular organelles. For the structure and classification of lipoproteins, and as depicted from the figure shown, lipoproteins are formed of a core of triglycerides and cholesterol esters, which are non-polar hydrophobic. So they are present in the core. And a shell that mainly contains phospholipids and some cholesterol, which are polar and confer some hydrophilicity. Embedded in this shell are protein molecules known as apolipoproteins, and they serve as regulators of lipoprotein metabolism. And to summarize, the inner core is formed of triglycerides and cholesterol esters, whereas the outer coat is formed of apoproteins or apolipoproteins, phospholipids, and cholesterol. For the categorization or classification of lipoproteins, lipoproteins are classified in different ways. They are categorized according to their size and density, which are denoted by their lipid and protein content. And as you see in the figure, the more the lipid content, the larger the size, and the lower the density, and vice versa. So the largest in size is a chylomicron molecule, which has the greatest lipid content compared to proteins or apoprotein content, and it has the lowest density. The smallest in size is the HDL, or high density lipoprotein, with the lowest lipid content and highest apoprotein content with the highest density. Regarding the functions of lipoproteins, as mentioned earlier, their structure helps keeping lipids soluble to facilitate their transport in blood. So they serve mainly as a vehicle to transport lipids from and to tissues. We have the protein components of lipoproteins, which is known as apoproteins. They represent the protein part or protein component of lipoproteins, and they are protein molecules weakly bound to the lipid component of lipoprotein and are exchangeable. These proteins serve different functions according to the type present in the lipoprotein. First of these functions is their structural role, as they are essential components of the lipoprotein molecules. Second, they act as activators of some enzymes involved in metabolism of lipoproteins and their mobilization. Third function is that they serve as recognition particles or recognition molecules to facilitate the uptake of these lipoproteins by specific tissues and cells. The plasma lipoproteins. As mentioned earlier, plasma lipoproteins serve as transport molecules for lipids, and we have two types. The triglyceride-rich type for the transport of triacylglycerols and include chylomicrons, responsible for transporting triglycerides of dietary origin. This means after meals, and they are synthesized in the intestine. Second is the very low dense lipoproteins, which transport triglycerides synthesized in the liver, or triglycerides of endogenous origin. Second part is the cholesterol-rich lipoproteins for the transport of cholesterol and includes the low density and the high density lipoproteins. Low dense lipoproteins transport free cholesterol and this is the most dangerous type of lipoproteins. Second is the high dense lipoproteins, which transport esterified cholesterol, and this is known as the good cholesterol. For the classification of lipoproteins, we have 5 classes of lipoproteins, 4 of which are outlined in the table shown. They all share the characteristic of containing cholesterol, triacylglycerol, cholesterol ester, phospholipids, and apoproteins, but at different proportions. They differ in the site of synthesis, the apoprotein component, and function. We have the chylomicrons that are synthesized in the small intestine, very low dense lipoproteins synthesized in the liver, low dense lipoproteins which are synthesized in the circulation and deliver cholesterol to tissues. Disturbances in the levels of low dense lipoproteins results in disturbed metabolism and atherosclerosis. The high dense lipoproteins, or HDL, which are synthesized in the liver and serve to deliver cholesterol from tissues to the liver. The fifth class or fifth category is the intermediate dense lipoproteins, which is between very low density and low density lipoproteins. The apoprotein component differs according to the type and function. Of lipoproteins, and as we see here in this table, for example, chylomicrons, the apoprotein component is apoprotein A, P48, C123, and apoprotein E. And as an example of their functions, apoprotein P48 serves for the assembly of apoproteins and lipids, and apoprotein C serves as activator, serves as an activator of enzymes and metabolism of this type of lipoprotein. The ApoP100 and the very low dense lipoproteins and low dense lipoprotein serves as a recognition particle for the very low density and low dense lipoprotein. They recognize the low dense lipoprotein receptors on cells. The lipid metabolism occurs in three major sites or three major areas. First of which is the intestine, where the chylomicrons are formed. Second is the extra-hepatic tissues, where lipoprotein lipase acts on very low density and low dense lipoproteins, and high dense lipoproteins are assembled. The last site is the liver, where very low dense lipoprotein is assembled and delivered to circulation, and intermediate density lipoproteins and low dense lipoproteins are metabolized. For the protein and their distribution and characteristics in lipoproteins. As we can see from the table shown, we have the apoprotein A1 and C2, which are activators of lipoprotein lipase, responsible for metabolism of chylomicrons to reduce their size. The B48, which targets the chylomicrons to the circulation and liver. The B100, which targets very low dense lipoproteins that contain triglycerides and cholesterol to tissues by binding to the low density lipoprotein and serves as a recognition particle for the receptor. And the ApoE or apolipoprotein A, which is responsible for targeting very low dense lipoprotein to tissues. And this slide summarizes the apolipoprotein distribution with the 4 main lipoproteins and their functions. As mentioned earlier, apolipoprotein A1 activates lecithin cholesterol transferase. The P100, which functions in triglyceride and cholesterol transport and serves as a recognition particle for the low dense lipoprotein receptor. The B48, responsible for transport of triglycerides. Apolipoprotein C2, which is an activator of lipoprotein lipase, and apoprotein E, which binds to low dense lipoprotein receptor. Regarding cells of chylomicrons and their assembly, chylomicrons are assembled in the small intestine, in the Golgi and the plasma reticulum of enterocytes, where the B48 apoprotein is added. Chylomicrons transport lipids after absorption to peripheral tissues after passing through the lymphatics, and its level is elevated 2 hours after meals. Very low dense lipoproteins are formed after lipoprotein lipase catalyzed removal of triglycerides from chylomicrons. They transport lipids from the liver to tissues. They have the P100 apoprotein, which is responsible for targeting very low density lipoproteins to tissues. This type of lipoprotein, the very low density lipoprotein, is converted to intermediate density lipoprotein, which represents the 5th group of lipoproteins. And finally, metabolized to low dense lipoprotein, which is uptaken by receptor mediated endocytosis in tissues, where its accumulation initiates or favor the development of atherosclerosis if deposited in blood vessels. As you see here, the very low density lipoproteins are converted to intermediate density lipoproteins and finally to low density lipoproteins, which are taken up by receptor-mediated endocytosis. The most dangerous one of these lipoproteins is the low-density lipoprotein, which originates by breakdown of very low-density lipoproteins and intermediate density lipoproteins with loss of the apoprotein C2 responsible for their metabolism and apoprotein E responsible for their targeting. The apoprotein B100 remains in these molecules and targets the low-density lipoprotein molecules to tissues. The last one is high-density lipoproteins, which mobilizes cholesterol from tissues and acidifies it by the lecithin cholesterol acetyltransferase activity of the ApoA1 apolipoprotein. This molecule transports the acidified cholesterol to the liver to be eliminated. We have two tables here that summarize the types of lipoproteins, their apoprotein content and percentage, their lipid content and percentage, and their origin and site of synthesis, as well as their functions. To summarize the process of lipid transport as shown in this figure, this figure shows transport and mobilization of lipids starting after absorption from the intestine. Chylomicrons are transported through the lymphatics by circulation to circulation. They are acted upon by lipoprotein lipase and changed into chylomicron remnants, which are transported to the liver and transformed into very low density lipoproteins. Very low density lipoproteins are targeted to tissues after losing some of its triglycerides and changed into intermediate density lipoproteins and low density lipoproteins. Then the low density lipoprotein is taken by tissues. Some of the cholesterol of low density lipoprotein is esterified to cholesterol ester by the L-cat activity and transported to the liver in the form of high density lipoprotein to be metabolized. The next point is Cholesterol. Cholesterol is an alcohol. It's a white waxy compound that is widely distributed all over the tissues with many important functions, of which is a structural function, as it is a structural molecule of cell membranes and cell organelles. Other functions include the bile acid and bile salts synthesis, steroid hormone synthesis, the recommended plasma levels of lipids, as you see here, for the total cholesterol is less than 200 milligrams per deciliter. Low density lipoprotein cholesterol, denoted as a bad cholesterol, less than 100 milligrams per deciliter. The high density lipoprotein cholesterol, or the good cholesterol, is about 40 milligrams per deciliter or higher, and triglycerides less than 150 milligrams per deciliter. Where the low density lipoprotein is associated with a high incidence of atherosclerosis and cardiovascular disease, and high density lipoproteins serving a protective role in the circulation. The last part is disorders of lipoprotein metabolism and mobilization. First, we have lipoproteinemia, hypolipoproteinemia, or the abetalipoproteinemia, a defect in loading the ApoB100 on the lipid component with accumulation of triglycerides in the liver due to defective triglyceride transport. Second disorder is a fatty liver, where there is increased hepatic sense of triglycerides and reduced mobilization of lipids from the liver. Second category of these lipoprotein metabolism disorder is the hyperlipoproteinemia. We have two important types. Type 1, hyperlipoproteinemia, due to lipoprotein lipase deficiency, which is responsible for the metabolism of lipoproteins. Second type is type 3 hyperlipoproteinemia, which is due to deficiency of the apolipoprotein E that serves as a recognition particle for the low dense lipoprotein receptors. There is also a high incidence of hypercholesterolemia and atherosclerosis with these two types. As you see here in this slide, these represent the laboratory findings in different types of hyperlipidemia as manifested by the turbidity of serum samples 12 hours postprandial. This table summarizes or lists some genetic causes of dyslipidemias that are considered as primary causes with defects in apoprotein synthesis or receptor defects. And finally, we have some causes of hyperlipidemias, which are secondary hyperlipidemias or secondary disorders, the most important of which are diabetes mellitus and hypothyroidism, which are associated with disordered lipoprotein metabolism and increased cholesterol and triglyceride levels in the serum. By this slide, we conclude the lecture for lipoprotein metabolism and their disorders.
Cholesterol metabolism. Topics included in this lecture are cholesterol metabolism, regulation, as well as hypercholesterolemia. What is cholesterol? Cholesterol is a white waxy substance. It's an alcohol with an OH group attached to carbon number 3. Carbon number 3, or the OH at carbon number 3, is the site of acidification with free fatty acids when cholesterol ester is formed. The sources of hepatic cholesterol. After lipid digestion, chylomicrons and other lipids are absorbed and bypass the liver to the circulation. The chylomicrons are processed in the circulation where they are converted into chylomicron remnants that are delivered to the liver, and this represents the first source of hepatic cholesterol. The second source is the reverse cholesterol transport via the high-dense lipoproteins, which take up cholesterol from tissues to the liver. High-dense lipoproteins take cholesterol from the chylomicron remnants and intermediate density lipoproteins. A third source for hepatic cholesterol is a de novo synthesis from the precursor substrate acetyl coenzyme A. So what about the fate of hepatic cholesterol? The very low density lipoprotein is synthesized in the liver from chylomicron remnants and is delivered to circulation where it is converted to the low density lipoproteins after binding to cholesterol and the apoprotein P100, which all form the transport vehicle of cholesterol to tissues. Part of cholesterol is excreted with bile to the gastrointestinal tract, and this represents a means of reducing the cholesterol level in blood. If bile becomes over or supersaturated with cholesterol, it may precipitate, and if infection occurs, this may lead to gallstone formation. This is all beside the sense of bile from cholesterol. So, these represent the phase of hepatic cholesterol. The biosynthesis of cholesterol, or de novo synthesis of cholesterol, starts with the substrate acetyl coenzyme A, or the active acetate. Active acetate comes from the oxidative decarboxylation of pyruvate, which is a result of glucose oxidation or glycolysis process. So pyruvate is oxidatively decarboxylated by the action of pyruvate dehydrogenase. Another source of acetyl coenzyme A is from the beta oxidation of fatty acids. So these acetyl coenzyme A molecules are delivered to the liver, where the cholesterol synthesis starts. Two molecules of acetyl coenzyme A condense by the action of a thiolase enzyme, forming acetoacetyl coenzyme A. And this occurs in the liver, placenta, intestine, skin, and suprarenal glands, which are the major sites synthesizing and utilizing cholesterol. The second step in cholesterol synthesis is the condensation of a third molecule of acetyl coenzyme A with the previously formed acetoacetyl coenzyme A molecule by an enzyme which is hydroxymethylglutarate coenzyme A synthase, followed by the rate-limiting step catalyzed by hydroxymethylglutarate coenzyme A reductase with the formation of mevalonate. This last step is the rate-limiting reaction of cholesterol biosynthesis, where pharmacological intervention can occur by statins that inhibit hydroxymethylglutarate CoA reductase and serve as a cholesterol-lowering drug. Mevalonate undergoes several reactions forming anosterol, sinosterol, desmosterol, and finally cholesterol. Talking about the regulation of cholesterol synthesis, regulation of the levels of cholesterol in the circulation as well as in the tissues occurs at different levels. The regulation of synthesis and uptake. As the cellular content of cholesterol increases, it exerts a sort of feedback regulation where the elevated cellular cholesterol itself inhibits the sense of the regulatory or key regulatory enzyme, the hydroxymethylglutarate CoA reductase. Also, there is reduction of low-dense lipoprotein receptor synthesis, and these two processes contribute to subsequent reduction of cholesterol uptake. A third effect is activation of an enzyme, acetylcholesterol acetyltransferase, which is concerned with cholesterol storage, whereas cholesterol is acetylated or esterified and stored in the form of cholesterol ester. Another level of regulation is exerted as a transcription level of hydroxymethylglutarate CoA reductase gene. And as mentioned earlier, serum cholesterol is a major effector of this process. The transcription of hydroxymethylglutarate CoA reductase gene is regulated by a protein SREP or steroid regulatory element binding protein, which is synthesized and activated in the endoplasmic reticulum and Golgi, then enters the nucleus where it binds to steroid response element on the DNA or the gene promoter, where transcription starts. And as shown in the figure, elevated serum cholesterol negatively affects the SREP, where SREP is bound to another regulatory protein, which is SCAP or SREP cleavage activating protein, leading to cleavage of SREP with the reduction of hydroxymethylglutarate CoA gene expression, or the HMG CoA reductase gene expression. Back to the regulation of cholesterol synthesis, we have the hormonal regulation. After meals, insulin induces activation of hydroxymethylglutarate CoA reductase by activating a protein phosphatase that dephosphorylates the hydroxymethylglutarate CoA reductase with subsequent activation and increased cholesterol synthesis. So, as we see, feeding promotes cholesterol synthesis by providing the substrates for cholesterol synthesis, which are acetyl coenzyme A and the reduced NAT phosphate, and activation of the regulatory enzyme hydroxymethylglutarate CoA reductase. During fasting and the periods of food deprivation, we have glucagon, the hormone that activates adenyl cyclase with increasing levels of the cyclic AMP and subsequent activation of a protein kinase that phosphorylates the regulatory enzyme hydroxymethylglutarate CoA reductase and inhibits its activity. Subsequently, cholesterol synthesis is reduced. This process is AMP dependent, so when ATP is reduced, there will be no need for cholesterol synthesis. So, as we see that fasting is a good cholesterol lowering strategy where the hydroxymethylglutarate CoA reductase activity is reduced. So, as you see, the covalent modification of the enzyme is key for regulating cholesterol synthesis. The hydroxymethylglutarate CoA reductase is covalently modified by phosphorylation dephosphorylation reaction. The active form is a dephosphorylated form. This is achieved by a protein phosphatase that is activated by insulin, whereas it is inactivated by phosphorylation by a protein kinase, which is activated by the hormone glucagon during fasting and starvation. And hence, this explains the role of fasting in down-regulating enzyme activity and cholesterol level reduction. To summarize the previous regulatory mechanisms for the activity of hydroxymethylbutyrate CoA reductase, we have short-term regulation by phosphorylation, dephosphorylation, and long-term regulation by proteolysis of the enzyme itself and cholesterol-stimulated degradation of the enzyme. Also gene expression and regulation by the steroid sensing domain and the strep protein. So, if we understood the previous points, we can now explain how does fasting inhibit cholesterol synthesis and explain how the feeding fasting cycle regulates the cholesterol metabolism. And one more question before the previous two questions. We can explain how carbohydrate-rich diet leads to cholesterol synthesis and hypercholesterolemia. So, if cholesterol synthesis is dysregulated, hypercholesterolemia may occur, and patients need to reduce their blood cholesterol levels pharmacologically by drugs. And as we know that bile acids, a great percent of bile acids is reabsorbed and recycled in the liver, where they have a great percent of the cholesterol with them, and only small percent passes with feces. So pharmacological intervention will block the recycling of bile acids and consequently forces the liver cells to utilize more cholesterol in bile acid synthesis, and this will share or contribute to the lowering of cholesterol levels in blood. The examples of these bile sequestering agents are cholesterol and cholestyramine, which are anion exchange resins, and as mentioned earlier, they act by blocking the intrahepatic recycling of bile acids and thus forcing the liver to utilize more cholesterol in sense of bile acids, and this will contribute to lowering of blood cholesterol levels. The bile acid sequestering agents are used in combination with other drugs, which are the statins, and statins are hydroxymethylbutyrate CoA reductase inhibitors. They reduce activity of the reductase, which is a regulatory enzyme for cholesterol synthesis, and this gives better results in treatment. As mentioned earlier, if dysregulation of cholesterol bile synthesis occurs, hypercholesterolemia may occur. An elevated cholesterol level in plasma above 220 milligrams per deciliter is considered as hypercholesterolemia, where the normal range lies between 150 to 220 milligrams per deciliter, and hypercholesterolemia, as we said, results from dysregulation of the mechanisms of cholesterol synthesis. This disorder may be familial or secondary to a primary disorder. The familial type may be due to a deficiency of the low-dense lipoprotein receptor, and this is a genetic defect of the low-dense lipoprotein receptor gene. And as we know, the low-dense lipoprotein receptor is responsible for internalization and uptake of the low-dense lipoprotein with its cargo of the high cholesterol content. Other causes or secondary causes of hypercholesterolemia include carbohydrate-rich diet, which provides the substrates for cholesterol synthesis. Carbohydrate provides acetyl coenzyme A and reduced NAD phosphate for the synthesis of cholesterol. A second cause is diabetes mellitus, where the lipid and all other types of metabolism are disturbed. The low-dense lipoprotein receptor and its metabolism are deranged in diabetics, and there is decreased clearance of the plasma lipoproteins, especially the low-dense lipoprotein with its high content of cholesterol. A third cause is hypothyroidism, and it is known that thyroid hormones stimulate bile acid synthesis from cholesterol. So these cases are associated with hypercholesterolemia and may be gallstone formation. Other causes may be some types of obstructive jaundice, which leads to decreased cholesterol excretion through bile ducts with bile acids. Also, obesity and nephrotic syndrome cases are associated with hypercholesterolemia, but the causes are unknown. Clinically, hypercholesterolemia may be manifested by hypertension, atherosclerosis, or atherosclerotic problems presented by myocardial ischemia. Other presentations may be with cases of hypothyroidism and their complications, skin manifestations as xanthomas of the eyelids, or eruptive xanthomas associated with hypertriglyceridemia. Also, it may be detected by routine laboratory investigations. Another problem that may be associated with deranged lipid and cholesterol metabolism is the fatty liver. The causes are summarized as overmobilization of fat and lipids to the liver, under mobilization of fat from the liver, or excessive carbohydrate intake, which leads to excessive lipogenesis and further accumulation of fats in the liver. This condition requires treatment. Treatment of the condition of fatty liver is in the form of agents that help mobilization of lipids out of the liver, and these are called lipotropic factors. And these factors are essential for the phospholipid synthesis, which are integral components of the lipoproteins responsible for mobilization of lipids and cholesterol and their transport across the circulation and tissues. These agents are in the form of choline and inositol. Some of these agents are provided by essential amino acids, and they are important for synthesis of choline, carnitine, and apoprotein synthesis. Also, essential amino acids as methionine and serine, and vitamin B12 and folic acid, which are important for the transmethylation reactions. How to treat hypercholesterolemia? The first line of treatment of cases of hypercholesterolemia are the lifestyle changes and dietary regulation. Also, pharmacological treatment by drugs as statins, which are hydroxybutyrate CoA reductase inhibitors. These drugs are structural analogs of the hydroxybutyrate CoA, which is a substrate for the enzyme, and thus inhibit cholesterol synthesis and reduce intracellular cholesterol. This will consequently lead to low-density lipoprotein receptor synthesis upregulation and increase uptake of cholesterol. And this will finally lead to lowering blood cholesterol level as we reduce intracellular synthesis and increase uptake from the circulation. Another agent is niacin, or nicotinic acid, which has a minor role in treatment of hypercholesterolemia due to its side effects and complications. Also, we have the oat bran, which are the soluble fibers of oat. These fibers bind bile acids with their cholesterol content, and this shares in lowering cholesterol blood levels. Other drugs used, as mentioned earlier, in combination with the statins are the bile acid sequestrants or bile sequestering agents, and their examples are cholestyramine, cholesterol, which sequester bile acids in the gastrointestinal tract and block their recycling. But these drugs also block absorption of fat-soluble vitamins. They may also bind some polar drugs as warfarin, digoxin, and thyroxine. So they have many side effects and drawbacks. So the best strategy for treatment will be the soluble fibers and the statins, as well as lifestyle change and dietary regulation. The last topic in this lecture is hypocholesterolemia, which is decreased level of cholesterol in blood below the normal level, which is 150 as a lower limit, 150 milligrams per deciliter. Some of the causes of hypocholesterolemia. First is a hyperthyroidism, which is a common cause for hypercholesterolemia. Other causes may be pernicious anemia, hemolytic jaundice, malabsorption syndrome, and other causes that will be discussed in a separate lecture. Thank you.
If you like this lecture, please hit the subscribe button and give your feedback in comments. and you can find other details for this lecture in the website provided.